Hey, so we're right in the middle of our cell division unit here, and we're looking at why and how cells do mitosis. And the first thing you got to know about mitosis is that you do cell replication for two reasons. Either you're growing or you're repairing. So that's the number one information that you always want to remember first. Growth and repair is all about mitosis. And so we're going to be looking at mitosis today on real cells. And when you start looking at real cells, it gets more complicated because we're not looking at animations on a computer anymore. We're not looking at perfect worksheets where you can see the chromosomes perfectly lined up or perfectly spreading apart. All right, so it gets a little confusing when we go to real cells. All right, so you need to know before we go into this lab, prerequisite knowledge here, you have to know your stages of mitosis. You have to know what interphase is, then what it looks like in prophase, metaphase, anaphase, and telophase. And once you think you got that, then we can look at real cells. Okay, so we're going to look at an onion root tip. Because if we're going to look at mitosis, we want to get some cells that we know are growing and dividing for repair and just for growth. And onion root tips grow really fast. So here is one onion root tip. On your slide, you're going to have three of these things, and they're lined up next to each other. Pick one. It doesn't matter which one. Okay? And when you see the whole root tip here, you want to get down to where you see the end of it is tapered, and then you want to back off a little bit. This is where the mitosis is happening the fastest. So this is where we're going to see the most stages of mitosis, all that PMAT that's going to be in here. So get it focused on low power. I'm on low power right now. Then, of course, once you get on low power, go to medium. I'm going to gain a little bit of magnification on you guys because I have the flex cam, and that adds a little bit of magnification for me. Let me get it focused in. It's going to be blue because these cells are stained. Of course, they're dead now, but this is like a little snapshot in time when this onion root tip was growing through the soil. All right, so I'm starting to see my cell walls. Those are all the lines going every which way. Those are all cell walls, and then the really dark stained blue things here, those are all nucleuses. All right, so when we go up to high power here, we're going to start to see what's going on in the nucleuses because that's going to give us our clues for what stage of mitosis we're in. Okay, high power, fine focus only. Go one way, got worse, go the other way. Let's see if I got a good thing here going on. All right, so I think I see everything that I need to see just in this one shot. Now, I did this beforehand, so I made sure that you guys were going to see everything you were going to need to see. You guys are going to have to hunt around a little bit for this. Your tools are going to be your two thumbs. So once you get on, I would suggest medium power, start looking around for an area where you start to see chromosomes doing something. Okay, you can't sit in one spot the whole time. You're going to have to hunt around. All right, so again, cell walls, we see the box, right? Here's another cell right here. And these are the nucleuses that we see in. Now we can see that not all things are going on the same in all of these nucleuses. So let's work our way up from interphase, and we'll go through the four stages so you guys know what you're looking for and what you're hunting for. All right, so interphase first. Okay, so here's a classic interphase right there. When we look at the nucleus, it's all unwound DNA. It hasn't formed chromosomes yet. So this cell is just living its life, being an onion root tip cell, doing all the functions that it needs to do to be an onion root tip cell. Most of its days of life are going to be spent in this category right here, which we call interphase. Then, at some point in the time, it's going to say, I'm going to divide for growth or repair. So the first thing that's going to do is it's going to get its chromosomes ready, which is going to take all this unwound DNA and it's going to wind them up. So I'm looking around here, I'm looking for a cell that looks like I can start to see chromosomes. And my eye comes right there. This is a good one of what prophase is going to look like. This one I would call a prophase right there as well. This one right here would be classified as a prophase. If we can see over here, that's a prophase too. These should be probably the easiest ones for you to find. I like to think of them as like little gummy worms. These are your chromosomes in there. And when they're kind of jumbled up in every which way, like that, like that, like that, that is a classic prophases. Okay, now they don't exist that way for long because now they're getting ready for metaphase. And always think meta in the middle. So now we're looking for chromosomes that are lined up in the middle. They're getting ready to split. I think I see 2 on here that I would classify as metaphase. Remember, this isn't perfect. These are living cells, or once living cells, so it's not going to be like a perfect animation. So I like this one right here for metaphase. You can see like the 50-yard line right here, and the chromosomes are going to be pulled apart here momentarily. I like this one is metaphase right there as well. And this one's close. It's not a great example, but it's close to metaphase. Okay, now the next one, this one's the hardest one to get because it happens the quickest. So remember, this is like a snapshot in time. So to get one that's an anaphase, where you're actually seeing the chromosomes being pulled apart, is the most rare, but you'll see them. This is a great one right here. So this is a classic example of anaphase. So you have these spindle fibers in here, which you can't see on this kind of a microscope, but they're pulling the chromosomes apart. And once they get pulled apart, then there's going to be another cell wall that forms right in the middle. That's called telophase, but we're not there yet, or telophase, depending on how you want to pronounce it. So here's a classic example of anaphase. Okay, now the next one is telophase. So now I'm looking for one that looks like it just split. The nucleus is starting to come back together around each identical copy of its chromosomes, and the cell wall is starting to form. These ones are tough. You kind of have to train your eye to see this. And I like 2 cells on here. I like these 2. I think these ones just went through an anaphase, and this one's nucleus is starting to get back together. This one's starting to get back together. And I like it because this looks like a new cell wall to me. So this looks like the old big cell here. They just split, and this is a new cell wall. I also like this one right here too, where this is the new cell wall, and they just split and went this way and that way. That one's a little tougher to see, but I've shown you all 4 examples. So again, interphase, 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 prophase, metaphase, anaphase, and telophase. All right, so focus up from low to medium to high. Remember, use your thumbs. Search all over that root tip, but stay mostly towards the tip because that's where the most growth is going to happen. Good luck and get your numbers counted.